Hello everyone, I'm Gong Yaoyang, an undergraduate student from PolyU ABCT. I will present our project on behalf of my team. As the title shows here, our team designed a peptide drug to treat triple-negative breast cancer through targeting autophagy. As we can see in the chart, the cancer which causes the most damage and has the highest infection rate for women is breast cancer. At the end of 2020, there were 7.8 million women alive who were diagnosed with breast cancer in the past 5 years, making it the world's most prevalent cancer. Breast cancer can occur in every country of the world in women at any age. Triple-negative breast cancer is the most difficult type to cure. 10 to 20% of breast cancer are triple-negative, and we do not have useful strategies to cure them. Therefore, the design of new drugs and tools are urgently needed. I'd now like to move on to introduce another term, autophagy. Autophagy is the cellular degradation and recycling process in our body for reusing old and damaged cell parts. Autophagy determines the fate of cells to survival or death. Our target is to design a new peptide drug to regulate cancer cells to undergo autophagy and lysis. Backlin-1 is a special protein which helps transfer our peptide drug, and then our drug will promote the autophagy reaction inside the cancer cells so as to achieve the purpose of killing them. Based on this mechanism, we have developed a novel peptide drug called TED-SP4, which binds to Backlin-1 and acts in cancer cells. These 6 graphs were drawn by us based on our experimental results, and all of them can prove that TED-SP4 has a high elimination rate of TNBC cells. The S-shaped curve on the left clearly shows that the number of cancer cells decreases dramatically in the presence of TED-SP4. Compared to other peptide drug TED-SP4, we found that TED-SP4 had a higher capacity to eliminate TNBC cells. From the line graph, we can see that TED-SP4 had a higher capacity to reduce the cancer cells, whereas TED-SP4 had almost no effect on cancer cells. Our in vivo studies also showed that TED-SP4 has a strong elimination effect on cancer cells and is nearly harmless to healthy animal cells. This is evidence that TED-SP4 has a strong medical value. And these shades are the detailed data obtained in our experiments. The future of TED-SP4 is certainly bright. Research on peptide drugs has been conducted for a long time in other countries and regions. Custom is an American bio company. Using this mechanism, they have successfully designed a drug for the treatment of lymphoma. They also raised $46 million in Series C funding last October. All of this goes to show that TED-SP4 is a world-changing technology. The TED-SP4 technology has been granted patent certificates in the United States and the People's Republic of China. We are now looking forward to financial support for our next step, safety evaluation. We aim to make the peptide drug to preclinical candidate within 2 years. This is the detailed schedule of our follow-up program. We plan to focus on the pharmacology of TED-SP4 over the next 2 years. And this is our team. Thanks for your listening. That's all my speech.